Hello students, welcome to CBSE NCERT Solutions by Vedantu. I am Miss Gopika, your own biology master teacher. So we have been doing NCERT line by line and the responses are really good on most of our videos, and we are super happy that students are enjoying this NCERT line by line. So today I'm going to do one of the most longest chapters, that is molecular basis of inheritance, and also a chapter that students find it very difficult to understand or decode. Okay, so I thought I will take this chapter up, molecular basis. We'll do a small portion today. We'll just do the introduction and try to understand what is this chapter talking about, or what is nucleic acid, and later we will learn more about this chapter. Okay, more about this chapter. Chalo. So molecular basis of inheritance, students, mainly stands on very simple concepts, that is your DNA and RNA. Okay, DNA and RNA. Now, who is DNA and RNA? DNA and RNA are none other than the nucleic acids. Okay, the nucleic acids. This is what they are. So, the whole chapter is based on this. The whole chapter is based on this. So, let's see it. Deoxyribonucleic acid, that is DNA, and ribonucleic acid, RNA, are two types of nucleic acids. Okay, two types of nucleic acid. So there was a scientist called Altman. Okay, he was the person who found out that actually it all started from Frederick Misher, who told that actually, the pus cells that he was working on, and let me tell you students, it was a mistake also, because he did not want to find this nucleic acid or nuclein or anything like that, but by fluke it happened, where Misher was able to find, where he was able to find something out of the white pus cells, something out of the white pus cells, and he noticed that, wait a minute, this is something that has a genetic information, or it can do something, was understood by Frederick Misher, and he found nuclein. But then another scientist called as Altman came, and he told that this nuclein is acidic in nature, so we will call it nucleic acid, and that is how the name nucleic acid came into existence. Okay, that is how the name nucleic acid came into existence. Definitely, history is not required for you, but it is good to know, right? This is how it happened and all of this, right? Awesome. So students, here they are telling that two types of nucleic acid are there found in living system. DNA acts as a genetic material in most of the organisms. RNA, though, it is also act as genetic material in some viruses. So DNA is the most common, prominent genetic material ever to exist. That is why common, I mean, is 90% or 99% of the organisms have DNA as the genetic material. The rest of it is RNA as a genetic material, especially in your viruses, like you have tobacco mosaic virus. All these viruses, you will see RNA as the genetic material. So they're telling in some viruses RNA is the genetic material. Mostly functions as a messenger. RNA has additional roles as well. It's not like just because RNA is not the genetic material, it does not mean RNA is useless. RNA has a lot of other purposes in the whole your central dogma, in the whole molecular biology. This is actually a part of molecular biology. It's like an introduction. So in the molecular biology, actually this RNA has a lot of additional job roles, right? Okay, it functions as adapter, structural, and in some cases as catalytic molecules. Okay, this we will go through in the later part of the chapters, but let's just try to read through. So in class 11 you have already learned the structures of a nucleotide. You have studied this, yes or no? You have studied this chapter you have studied it in biomolecules, the structure of a nucleotide. You have studied about purines, pyrimidines, a little bit you have gone through it. Okay, so that chapter is like a foundation for this chapter, and the way these monomer units are linked to form the nucleic acid polymers. In this chapter we are going to discuss the structure of DNA and its replication, the process of making RNA from DNA, the genetic code. So they have just told what and all you are going to learn in this chapter, that is so important. Okay, so important that you cannot even think of skipping it. Okay, you cannot even think of skipping it. And let me tell you students, for NEET students, this chapter is very important, and for both students, let me tell you, all the teachers love to give questions from this chapter, because this chapter can easily every teacher blindly can make like so many questions out of it. Okay, every teacher can blindly make so many questions out of it. So this chapter is everyone's favorite chapter. Okay, be NEET, it is like NTA's favorite chapter, because around 40 questions we can expect from this chapter. Okay, around 10 questions you can expect from this chapter. Goes in a variation of from 8 to 10 questions you can definitely expect from this chapter. Okay, so let me just give you an introduction to the chapter in my own way, because see, even though I'm doing NCERT line by line, I always make sure that I give you an introduction from my point of view. So this way what happens, students find it very easier, like, okay ma'am, we can connect. Okay, so yes, so the introduction is that this chapter, the entire chapter, if I have to talk, it is talking about the genetic material. What is it talking about? Genetic material. Now, what is genetic material? What is this molecular basis of inheritance? Inheritance means what, students? Now, this is your grandfather. Okay, this is our grandfather. Okay, this is the grandfather. He had a plot. Okay, he had a plot. Now, this plot is inherited to your father. Okay, to your father. Okay, now from your father, that same plot, okay, which your grandfather gave, your dad did not sell it, so that plot is now on with you. Okay, now with you. Now what is this? This property that your grandfather had, okay, the what your grandfather had was inherited by your father, and now it is inherited by you. Okay, but see, imagine your grandfather did not sell any portion of the property, but your father sold a small portion of the property. Now when you are inheriting it, right, when you are inheriting it, you have that much less. Okay, you have that much less of the property. Now what is going to happen? That is, the property, the inherited property that came from your grandfather is, not completely inherited to you. But when it comes to our body, okay, when it comes to our body, that when an information from your parents, from your maternal and paternal side has to be inherited to you, okay, it is made sure that a genetic, like something who is carrying this information from your parents to you, has to be a very reliable source, or very, a kind of source that will not leak any information. Like how you saw, no, from your grandfather, the entire property came to your father, but your father sold a small portion of it. Okay, and then you got the rest of it. That is what the information is broken, or the information is leaked, or there is a change in what is inherited. But in our body, that will never happen, because the genetic materials are so well, maintained, or the entire information will be inherited from you, from your parents to you, from you to your kids, right? So it will go generation after generation. So genetic material is not a simple thing, it is a very necessary thing, and all living organisms have it. Okay, all living organisms have it. Got it? Now let's say, what is the next topic, ma'am? When we talk about genetic material, we have to talk about nucleic acids, because nucleic acids are the one who are forming this genetic material. Yes or no? So nucleic acid, students, are of two types. Okay, mainly two types. I mean, not mainly, just two types. One is going to be your DNA, one is going to be your RNA. Now ma'am, what is this DNA? DNA means deoxyribonucleic acid. Deoxyribonucleic acid. Okay, RNA means ribonucleic acid. Now deoxyribonucleic acid means deoxy. From the term itself, you understand that something is missing here that is not missing here, right? So deoxy means what? This person lacks one oxygen. Lacks one oxygen compared to your RNA. And let me tell you, lack of this one oxygen is not a simple thing, because lack of this one oxygen makes this entire DNA very stable, structurally different, all of this. Okay, this one oxygen which is extra in this RNA makes this RNA more reactive. Okay, more reactive. Is this clear to all of you? Yes or no? Okay, voice is also clear. Yes, voice is clear, I guess. Okay. Oh no, I'm sorry. Yeah, voice is also clear, all of you. Any doubts? There shouldn't, like, so far, if you have any doubts, definitely drop it in the comments. Do not hesitate thinking that, oh, ma'am might not see the comment. No, we will see the comment. Okay, chalo. Now students, a little bit about the history of DNA. Okay, a little bit about the history of DNA. History of DNA. So history of DNA, as I told, okay, history of DNA, as I told, it was none other than Michel. Okay, Frederick Michel. He was working on pus cells. Okay, he was working on pus cells. Excuse me. Okay, he was working on pus cells, and when he was working on pus cells, he happened to find something called as nuclein. He found something called as nuclein, and this was the first ever introduction to nucleic acid. Then another scientist came called as Altman. Okay, Altman, and he told this nucleic acid has an acidic property. What? An acidic property. Because of this acidic property, why don't we call this as nucleic acid? Why don't we call it as nucleic acid? And that is how the name nucleic acid came into existence, and then the DNA and the RNA. Now, one more thing that they are mentioning definitely in the chapter is that DNA is a genetic material. Okay, DNA is a genetic material for who? For most of the living organisms. Okay, most of the organisms, your DNA is the genetic material. Got it? Most of the organism, DNA is the genetic material. But what about RNA, ma'am? In viruses, RNA is the genetic material. Not all viruses, but in most of the viruses, RNA is the genetic material. Okay, we can take example of TMB. TMB. Got it? Yes. This is the introduction. Now let's go further and let's understand what are we learning in this chapter. Okay, what are we learning in this chapter? So look here, this chapter, this paragraph, I'll come back to it. So in this chapter, you are going to learn about obviously the nucleic acids. Okay, nucleic acids, and you're going to learn a lot about DNA and RNA. Okay, then you're going to learn about how DNA is formed. Okay, how the nucleotide is forming the chain. Okay, how they are double helical structure, double helical, okay, helix structure. Then you will learn a lot about the central dogma, how the whole, how is the information passing to us, or how are we inheriting each of the characters. You learn a lot about that. You learn a lot about different experiments that was done. Okay, different experiments that was done to make sure that our DNA was the genetic material. Then you will learn about how they found the biochemical nature of them. Okay, biochemical nature. Okay, and much more. This is what the agenda of this class is, but you have a lot to learn. Okay, like transcription, translation, but let me tell you, it is very easy. Okay, it is very easy. You do not have to worry even for a minute that, oh my god, it's so difficult, will I be able to learn, I don't know anything about this. Don't worry at all. This is very easy topic. Okay, very easy topic. You'll be able to learn. Got it? Yes, all of you. Okay, fine. Oh, students, did the video go? I'm so sorry. Okay, this is the thing. Okay, this is the whole chapter. Now, in the last section, the essentials of human genome sequencing and its consequences will also be discussed. Now, let us begin our discussion by first understanding the structure of the most interesting molecule in living system, that is your DNA, and the subsequent sections, and we will understand why it is most abundant genetic material and what is its relationship with RNA. Okay, this is the introduction of the chapter. Now let's understand DNA. So before we go into understanding the DNA, you have to know the how many nucleotides are present, what is there, all that's a few numbers that you have to know. See, DNA is a long polymer of deoxyribonucleotides. Obviously the name of DNA is deoxyribonuclease, so obviously it will be called nucleotides. Okay, the length of the DNA is usually defined as the number of nucleotides. So if I have to tell about the length of DNA, okay, length of DNA, that means it is the number of nucleotide. Okay, it is the number of nucleotide. Now, or a pair of nucleotide refers to as base pair. So a nucleotide can be single, or there can be a pair, which we call it as the base pair, which you will study later. So this chapter assumes that you know biomolecules, and it is not the first time that you're listening to these terms. But let me tell you, if you're first time learning molecular basis of inheritance, skipping biomolecules, then there is no use at all, because if you don't know biomolecules, some of the terminology, some of the bondings here is going to be difficult. Okay. Now, this is also characterized of an characteristic of an organism. For example, a bacteriophage. Okay, a bacteriophage is a virus that can infect bacteria. Is known, okay, is known as 5174, which has 5386 nucleotides. Okay, which has 5386 nucleotides. Got it? These numbers you have to by heart. There is no hook and crook to it, and many people make, you know, examples telling 7 plus 4, 7 minus 4, 8 and all that, but that is too complicated. Students, just go for, if you write it 2 times, you're going to remember. Okay, 5174. Okay, 5175 174 is going to be 5386. Okay, next one, you have a bacteriophage lamla, which is going to have 48502. Okay, 502 base pair. Can you see here? This is nucleotide, this is base pair. Okay, that means this is indicating that I am a single strand. Okay, I am a single person, but here I am indicating that I am paired, or I have a base pair. Understood? So this differences you should understand. This differences you only understand if you know biomolecules. Okay, so you should definitely watch biomolecules lecture before watching molecular basis of lecture. Okay, now bacteriophage lamla has 48502 base pairs. Now E. coli has 4.6 into 10 to the power 6 base pair. So even in E. coli, the base pairs are paired, right? Clear? Yes, the base pairs are paired. Now, and haploid content of human DNA. Student, this word is very important. Haploid content of human DNA has 3.3 into 10 to the power 9 base pairs. That means diploid is going to be 6.6. Okay, diploid is going to be 6.6. Okay, haploid is going to be like in your sperms and eggs, other things, but diploid content is going to be 6.6 into 10 to the power 9 base pair. So do not make this mistake when you are answering somewhere question that, oh, they told haploid, but then the question was diploid. You should basically do it into 2, 6.6 into 10 to the power 9 base pairs. Okay, here they very clearly mentioned haploid content of a human DNA. So students, these 4 things you have to know it by heart. There's no other choice. Okay, be it 5174, it is 5386 nucleotide, be it about E. coli, be it about lambda bacteriophage, everything you have to know. Okay, everything you have to know. Chalo. Now let the structure of, one second. Okay, let's study the structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain. A nucleotide has 3 components: a nitrogenous base, a pentose sugar, and a phosphate group. There are 2 types of nitrogenous base, purines and pyrimidines. Okay, purines are adenine and guanine, pyrimidines is cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present only in DNA, uracil is present only in RNA. Okay, a nitrogenous base is linked to the OH of the first carbon pentose sugar through a N-glycosidic linkage. So they're trying to say that nitrogenous base, when it binding with sugar, it binds with a N-glycosidic linkage. Okay, N-glycosidic linkage. Okay, from the to form a nucleoside, such as adenosine, deoxyadenosine, guanosine, deoxyguanosine, cytidine, deoxycytidine, uridine, and deoxythymidine. Okay, or deoxythymidine. When a phosphate group is linked to the OH of the 5th carbon of a nucleoside to a phosphodiester linkage. So a phosphate is going to link to a nucleoside. Okay, a phosphate is going to link to a nucleoside with the help of a phosphodiester linkage. Clear? Okay, now I will definitely explain it. Don't worry. I'm just asking you, like, if you understood the lines of NCERT. Okay, lines of NCERT. Now we have to go little in detail about the components of nucleotide. Okay, let's do that. Okay, so students, when we start, we should understand that DNA is made up of a polynucleotide chain. Made up of a polynucleotide chain. Now, polynucleotide chain means what? One linked with another one, linked with another one, linked with another one. They make a long chain. But whom are they linking? Who is linking? Okay. Now, this polynucleotide chain is made up of a single unit of nucleotide. Yes or no? It is made up of a single unit of nucleotide. Now, this nucleotide has three components. Okay, this nucleotide has three components. What are that, ma'am? They have nitrogenous base. Okay, nitrogenous base, pentose sugar, and phosphate group. And phosphate group. Pentose sugar and phosphate group. These are the three components of your nucleotide. Now, each of the components, let me say, you do not have to know a lot about it, but definitely you have to know some amount of it. Okay, some amount of it, because I'll tell you why. Because these topics have been in biomolecules, but still you have to know some things about this, so you understand. Okay, where is the bonding happening, what is happening, all that you will be able to understand. Got it? Chalo. Okay, now we will start with nitrogenous base. Nitrogenous base. Now, ma'am, what is nitrogenous base? Students, nitrogenous base is made up of two components. Okay, called as purines and pyrimidines. Purines and pyrimidines. Okay. Purines are made up of two components called as adenine and guanine. Okay, guanine. Got it? Now, pyrimidines are made up of three components. Okay, pyrimidines are made up of three components. You can remember it as cut. Okay, cut. That is cytosine, uracil, and thymine. This is the three components of pyrimidines. Now, remember that pyrimidines are a 6-ring structure. 6-ring structure. 6-ring structure means like this. 6-ring only, no? Yeah, like this. Okay. 6-ring structure. Always confused about chemistry little. Okay, 1, 2, 3, 4, 5 and 6. It's a 6-ring structure. But your purines are made up of 9-ring structure. So students, nothing is there in name, because don't judge a book by its cover. That is the answer here. Don't judge a book by its cover. Why I'll tell you, because see, purines is a small name, right? Purines is a small name, but pyrimidine. Okay, pyrimidine is a long name, but still, what has happened? This people's name and the ring, right? Name and the ring, it does not matter at all. Okay, does not matter at all, because the purine is a small name. Okay, purine is a small name, but the ring is 9-ring structure. Pyrimidine is a big name, but it is only a 6-ring structure. So do not judge a book by its cover. This is the definition of it. Okay, do not judge a book by its cover, because if I have to draw a purine, okay, I don't know if I will fit it, if I can fit it here. So you can draw a 6-ring structure. Okay, 6-ring structure, and then you draw a structure like this. Okay, like this. Okay, now you can start labeling it, like numbering it. What I'm trying to say is numbering it. Okay, so 1 second here, I think it's too small also. Okay, too small, so we start from here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 7, 8, 9. 6, 7, 8, 9. So 9-ring structure. Okay, 9-ring structure. Now there's a lot about purine and pyrimidine actually. I think, where do I write it? Yeah, I'll write it here. Okay, write it here. What is purines and pyrimidines? See, remember students, this purines and pyrimidines, no? Now if the DNA structure is like this, okay, this is the DNA. Okay, it's a double helical structure, no? Like this the DNA is. Now this is the backbone of the DNA. The white color one is the backbone of the DNA. But who is making sure that this backbone is going to stay? That is going to be your purines and pyrimidines. So they will project outside like this and they're going to bond. So remember that adenine, okay, adenine will bind with thymine, bond with thymine. Same way, cytosine will bond with guanine. I make a trick to remember this at Gopika's class. Why? AT, adenine thymine, GC. Okay, Gopika ma'am's class. Now, when I drew it also, you would have noticed that I did not do a mistake, but I just drew here two bonds, two hydrogen bonds, and here I drew three hydrogen bonds. Three hydrogen bonds. Why so? That is because cytosine and guanine, or guanine and cytosine, is linked with each other with three hydrogen bonds, which is more strong. Okay, strong bonds. This is more strong bonds, and here this is two hydrogen bonds, which is a little more weak bonds. Okay, now remember that they had told some things about bonding, right? So if you if you want me to draw the bonding, then it is okay, and then it is going to be different. So okay, it's okay. I will just draw about the sugar. Okay, I'll just talk about the sugar. Now, students, bonding we will come back to, because I feel now if I talk about bonding, it might be confusing for you. So so adenine, thymine, cytosine, guanine, I hope all of you understood. Now you will be thinking, ma'am, uracil you left. Yes, because uracil is present only in RNA, and it is a replacement for your, okay, replacement for thymine. A replacement for thymine. Or a replacement for thymine. Now students, remember, okay, remember, next component was what? Nucleotide is made up of three components: nitrogenous base, pentose sugar. Okay, pentose sugar. From the term itself you understand, no, sugar, and it is pentose, that means 5 carbon sugar, right? 5 carbon sugar. Obviously in DNA it's going to be deoxyribonucleotide. Deoxyribonucleotide is the one. Okay, now it is deoxyribo sugar. Okay, let's write sugar, ribo sugar. Now they are telling that the carbon is a 5-ring carbon. Okay, 5-ring carbon. So we have to draw the structure. This cyclic ring and non-cyclic ring, we have to be careful, because even one arrow goes that side, I mean one line goes that side, it is going to go wrong. Okay, this is a sugar molecule, students. Now here carbon molecule will be there, and this is going to be the third one. You see, look here. So 1, 2, 3, 4. Okay, then where's the fifth carbon, ma'am? This is the fifth carbon. Okay, this is the fifth carbon. This is a deoxyribose sugar, because here I'm only going to write H. Okay, there I'm only going to write H. No, second carbon, sorry. Okay, second carbon we will write a H. Second carbon is going to have only a H. So this is going to be a deoxy one. Okay, and the other one, this one, will be a ribose sugar. Same thing, students, everything is same, just that on the second carbon, here it is going to be an OH group, here it is going to be an H group. This is going to be ribose sugar. This is clear? Ribose sugar. So now we did nitrogenous base as well as pentose sugar. Then phosphate, there is nothing much for you to remember. You just have to remember phosphate group is present. Okay, phosphate group is present to look like this. Okay, phosphate group is present. This much only you remember. Okay, this much only you remember. A lot of chemistry is there in your biology, so that's why don't take tension. Oh my god, ma'am, what have you drawn and kept? Don't take tension. Okay, it is going to be easy. Okay, it is going to be easy. We'll make sure that you understand also. You understand as well. Got it? Okay, fine. Students, if you want proper notes of it, you can join my Telegram, Biology by Gopika, where there is a beautiful notes of your biomolecules, where I have drawn the structure of each diagram and explained, so you can take that notes as well for revision. Okay. Now, students, here what did they tell? They told that when pentose sugar and nitrogenous base combines. Okay, now let's see here. When they, so listen to this carefully, nitrogenous base plus sugar combines, it will form, okay, this will form a nucleoside. That means pushing one person to the side. Whom are they pushing to one side? The phosphate group. Whom are they pushing to one side? The phosphate group. So phosphate group is not included. So whenever a nitrogenous base and a sugar has to make a bond, it is called as N-glycosidic linkage. Glycosidic linkage. Got it? N-glycosidic linkage. Now when they have to combine this phosphate with the nucleoside, so nitrogenous base plus sugar is equal to nucleoside. Now when this phosphate group has to combine with the nucleoside, they are going to bind with a phosphoester bond. Okay, phosphoester bond. Very simple, students. See, glucose, glyco, sugar. Okay, glycosidic linkage. Phosphate, phosphoester bond. Okay, phosphoester bond. This if you remember, then everything else is easy. See, this if you remember, everything else is going to be easy. Got it? Now they're saying this bonding is clear, no? Now here, when the phosphate group, okay, when the phosphate group is linked to OH of the fifth, it will form a phosphodiester linkage. A corresponding nucleotide or a deoxynucleotide, depending upon the type of sugar, is present or formed. Now look here, they are telling two nucleotides are linked to through 3' to 5'. Okay, through two nucleotides are linked to 3' to 5' phosphodiester linkage. That is when, see, now this is this one. This carbon, when let's say this, okay, when this one is binding with this phosphate, it is going to form a phosphoester linkage. Yes or no? Phosphoester linkage. But when this and this is binding, phosphodiester linkage. That means two nucleotides are binding together, phosphodiester, single nucleotide ester. Okay, ester. Then a polymer thus are formed at one end, a free. So here, students, if you see, there is a free phosphate group. There is a free phosphate group, and here there is a free or hydroxyl group. So this is going to be the 5' end, and this is going to be a 3' end. Why? Because here there is a 5' phosphate free, here there is a 3', at the 3' there is one OH free. So that is how we tell 5' to 3' and 3' to 5'. This is how we understand the polarity of the DNA. Okay, now look here. Phosphate at the 5' end of the sugar, which is referred to as the 5' end of the polynucleotide. Similarly, at the other end of the polymer, the sugar has a free OH at the 3' end of the group, which is referred to as the 3' end of the nucleotide. Okay, the backbone of nucleotide is formed due to sugar and phosphate. So as I told you, the sugar, don't get it confused. Okay, don't get confused with nucleoside and backbone. Sugar and phosphate is going to form the backbone, and the nitrogenous base are going to project inwards. Okay, they are going to project inwards and make sure that structure stays intact. Okay, the double helical structure is formed. Not only formed, students, to keep it in place also we need this people, no? Because there are two strands like this. To keep them in place, we need like this bonds to hold them. So adenine will bind with thymine with 2 hydrogen bonds. Guanosine will bind with cytosine with 3 hydrogen bonds. Okay, the backbone of polynucleotide chain is formed due to sugar and phosphate. The nitrogenous base is linked to sugar moiety projects from the backbone. Okay, backbone. Now look here. I think I should have left more blanks. Okay, I have to teach you some more things. Okay, now students, I'm going to draw a purine. Okay, I'm going to draw a purine. It's okay, we have slides in the last, we can use that slides. See, this is a purine. Okay, yeah, this is a purine. So 9, 8, 7, 6, 5, 4, 3, 2 and 1. You'll be like, ma'am, how are you labeling? If Diksha ma'am sees, she's going to cry. Yeah, so like, you know, some jugaad will be there, no, for us to remember. So students, on purine, okay, on the 1, 3rd, 7th and the 9th, there is going to be a nitrogen group present. All the odd numbers. Okay, 1, 3, 1379, you're going to see a nitrogen group. Okay, then on the pyrimidine and on the first and the third position, there is going to be a nitrogen group. Now look here, this is the ninth position. Now actually I should have drawn this down, but just to show you, I'll draw it here. Okay, this is actually not right because it should have been up. Okay, because what I wanted to show is this and this making a bond. So I'll do one thing, this I'll draw it this way, this side I'll draw it. Okay, what I'm drawing is a sugar. So students, if this is the first, okay, 1 dash, 2 dash, 3 dash, 4 dash, and your CH2OH is going to be the 5 dash. Now you can ask me a question, ma'am, why are these people 5 dash, why are they 1, 2, 3? To differentiate each of them, no, they have put a dash for the glucose or the sugar groups. Otherwise, what is going to happen? We will not know with who is binding, right? So students, whenever a sugar is binding with a nitrogenous base, it is going to form an N-glycosidic linkage, we told, no? So now between a purine and a sugar, it is going to be 1-9 N-glycosidic linkage. Okay, N-glycosidic linkage. Got it? 1 comma, see this to understand this difference, that okay, on the first carbon the binding is happening, and on the ninth purine, okay, on the ninth nitrogenous base it is happening. We have to differentiate with this dash. Okay, 1 comma or 1,9, you can tell 1,9 N glycosidic linkage is formed. Is this clear, all of you? Yes. Okay, so now with the third carbon, okay, with the third carbon, now what I'm drawing, please don't think that, oh my god, what has ma'am drawn and kept. This is just to show you, okay, is going to combine with your phosphate, right? It's going to combine with your phosphate. So this is all like, you know, Jugaad diagram, just to make you understand, but when I draw it properly, it is different. Okay, so it will bind with this one and form the phosphoester bond. Okay, phosphoester bond. This you just remember. Okay, this you just remember. Possible? Okay, so this, what is going to happen, students? This oxygen is going to shift to the center here. Okay, the whole line will event. So here O group, and it is going to bind here. Okay, it's going to bind here. Yes, so this is how the binding happens. Okay, from purines. Now this is the purine. So obviously 9-ring structure. On the ninth position binding is going to happen. Here it is a sugar and here it is a phosphate group. Okay, this is how it is. Now further we are going to study about how the double helical structure and all came. So let's go back to that. So here, okay, in RNA, every nucleotide residue has an additional OH group. Every nucleotide residue has an additional OH group present at the second position in the ribose, which I showed you. Also in RNA, the uracil is found at a place of thymine. Okay, 5-methyl uracil, another chemical name for thymine. Okay, it is present where, students? Uracil is present in DNA and in RNA, and thymine is present in DNA. Now DNA as an acidic substance present in the nucleus was first identified by Friedrich Mischer in 1869. Okay, this date you remember, 1869. He named it as nuclein. However, due to the technical limitation in isolating such a long polymer intact, the elucidation of the structure of DNA remained elusive for a very long time. That is, they were not able to understand what is the structure of DNA, how do they look like, what is happening. They were not able to understand that. Okay, clear? Just to give me 1 minute. Okay, fine. Okay, DNA remained elusive for a very long time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction, okay, on the X-ray diffraction data produced by Morris Wilkins and Rosalind Franklin, understood that the structure was double helical. So students, they viewed the DNA structures under the X-ray diffraction, and they noticed that DNA is a double helical structure. But who was the first person to find this? It was not Watson and Crick, it was Wilkins. Okay, it was Wilkins and Franklin. Now Wilkins, Hershey Chase, that is, sorry, Watson and Crick. Okay, Watson, Crick. This three people got recognition for this, but Franklin had passed away by that time, so that is the reason Franklin did not get the Nobel Peace Prize. Okay, so here, I mean Nobel Prize. So here, double helical model of the structure for the DNA. One of the hallmarks for their proposition was base pairing between two strands of the polynucleotide. However, this proposition was based on the observation of Irwin Chargaff rule. So we'll come to Chargaff rule. So students, here this whole diagram, this whole paragraph is about how they found the nuclein, which I already taught you. In 1869, Frederick Mischer found it in pus cells, and he called it as nuclein, and Altman changed it to nucleic acid. But the structure of the DNA was not not known to us, so they kept finding. Or later, Wilkins and Franklin did an X-ray diffraction separately. They did, and when they saw the X-ray diffraction, they noticed that DNA is a double helical structure. And later, Watson and Crick also gave the name, telling that this is a double helical structure and these are the properties of this double helical structure. And then Irwin Chargaff came up with Chargaff rule. Okay, came up with Chargaff rule. Now, what is this Chargaff rule? It's very simple. Okay, Chargaff rule. Now, what is this Chargaff rule, students? Very, very simple thing about Chargaff rule is that, see, they told that if there is adenine, okay, adenine will bind with thymine, and this you already told us. Very simple. Let's consider this is a purine. Not let's consider this is a purine. This is a pyrimid. So the ratio of purine is always going to be equal to the ratio of pyrimidine. Yes or no? If in a class 10 students are there, I will tell make a group of 5, you will make a group, 2 groups you will make. Same thing reverse, if I ask you, if I tell you make a group of 2, you will make 5. But if it is 11 students, then one student is going to be left out, right? So I should always make sure that the ratio of boys and the ratio of girls is equal to make sure that the groups are formed, or the ratio of girls and the ratio of the students in the class should be equal, so that I can make a group. Same thing. How much pyrimidine is there, that much purine should be there. How much purine is there, that much pyrimidine is there. Otherwise, whom will they bind to? Right, adenine has to bind with thymine. So if adenine 10 is there, even thymine 10 should be there. Otherwise, adenine will be alone. Same way, if cytosine 10 is there, guanine also 10 should be there. So here, Chargaff called his rule as Chargaff rule. He telling that ratio of purines will always be equal to the ratio of pyrimidines. But there's also a thing that AT and GC need not be equal. That means what? The amount of adenine and thymine. The amount of adenine and thymine need not be equal. Okay, need not be equal to guanine and cytosine. Guanine and cytosine. Yes or no? Why should these two be same? Okay, this is one pair, this is another pair. Do these pairs have to be same? Not required. Okay, so many students have this notion that, okay, adenine, thymine is equal, guanine, cytosine is equal, so these all will be in same number. No, students, there are regions in the DNA where we have guanine and cytosine rich regions. There are regions in the DNA where we have adenine and thymine rich regions or less regions. So this can keep changing. Okay, this can keep changing. Maybe guanine and cytosine is 60% there in DNA, but this is only 40% there. It's possible, completely possible. But adenine should always be equal to thymine and guanine should always be equal to cytosine. Got it? So he told the rule called as Chargaff rule. That is, the base pairing confers a very unique property of nucleotide to the nucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence of the other strand can be predicted. That is, they are complementary to each other. That is, see, if I know that it is A, okay, GATTC, then I can definitely write what is going to be in the other strand. So this strand is 5 dash to 3 dash. The other strand which is going to be formed, okay, which is going to be formed, that is 3 dash to 5 dash. What is it going to be? A T T C T A G. So he told that these people are going to be complementary to each other. Okay, complementary to each other. Clear or not? So this is a one strand. If I know another stand also, I will be able to know. Okay, I'll be able to know. So students, I'm very sorry, my allergies are not reducing at all. Okay, I have extreme dust allergy, and that is why every day you will be like, why is this ma'am always having this throat, and the entire face will be swollen. It is because of my allergies, so please excuse me for my voice, and sometimes there's going to be runny nose and all of that. So just understand that ma'am has an allergy. Okay, so yes, also if each strand from the DNA, let us call it as the parental DNA, act as a template for the synthesis of the new strand, the double stranded DNA, let us call them the daughter DNA, thus produced would be identical to the parental DNA because of the genetic implication. So what they're trying to say, they're trying to say that if, let's say, this is the parent DNA, okay, this is the parent DNA. If this parent DNA is AGAATTC and the parent DNA is making a copy of the daughter DNA, okay, this is going to be the daughter DNA. This you'll study more about it in replication. Daughter DNA, what is it going to be? T C T T A G. Yes. So they are telling that if the parent DNA stands as a template and forms the daughter DNA, they both are going to be complementary to each other. Okay, they are both going to be complementary to each other. But remember that we'll only be taking one strand of the parent DNA, right? Because if we take this strand of the DNA and this strand of the DNA, same way for parents also, then they are all going to be complementary to each other. So if we take one strand of the DNA, this both are going to be complementary to each other. No doubt in that, because one strand is complementary, another strand also will be complementary. Okay, this will be 5 dash to 3 dash direction. So this is followed throughout the DNA structure. So what and all did you study, students? You learned about Chargaff rule. Okay, Chargaff rule, very important. You learned about how the double helical structure, okay, double helic structure is found. Okay, found, or yes. Now the salient features of double helical structure of DNA. Very simple salient features. Okay, students actually complicated by studying, but let's see the salient features of double helical structure of DNA is that it is made up of two polynucleotide chain. This all of you know now. Many nucleotides combine to form the polynucleotide chain, okay, where the backbone is going to be sugar and phosphate and the bases are going to project inside. Okay, the two chains have anti-parallel polarity. The two chains have anti-parallel polarity. That means what? If one is going from 5 dash to 3 dash, the other one will go from 3 dash to 5 dash. This is also because of the free ends, because here there is the phosphate group free end, here there is an OH group free end. Same way, this will keep continuing, okay, when the binding happens. It means that if one chain has polarity 5 dash to 3 dash, the other will have 3 dash to 5 dash. The bases in two strands are paired through hydrogen bonds, forming the base pairs. That is, adenine will bind with thymine with 2 hydrogen bonds, and guanine will bind with cytosine with 3 hydrogen bonds. Okay, this is the next thing. And here you can also write about the Chargaff rule. Okay, now the two chains are coiled in right-handed fashion. Okay, two chains are coiled. That means this is one chain, this is one chain. They are coiled in a right-handed fashion. Okay, the pitch of the helix is 34 nanometer. Okay, 34 nanometer. So the turns in the DNA going to be 3.4 nanometer. okay, and they are roughly, that is 10 base pairs will be there in one turn of the DNA. Okay, if this is, let's say this is one turn of the DNA, in this one turn you will spot 10 base pairs. 10 base pairs. Okay, 10 base pairs will be seen in one turn of the DNA. Friends, here you can see, okay, this is the bonding that we spoke about. So phosphate, sugar, you know the binding, or sugar and nitrogenous base. First, this binding you can remember with N-glycosidic linkage. Okay, glycosidic linkage. Here you can remember the sugar. This one is going to be with phosphoester bond. Okay, phosphoester bond, and these are going to be the hydrogen bonds. These are going to be the hydrogen bonds. Got it? Hydrogen bonds. Okay, you can see hydrogen bonds here. Here 2 is there, here 2 is there, here 3 is there, here 3 is there. Now, so here 2 more points are there, students. Yes, so consequently the distance between the base pair and a helix is approximately 0.34 nanometer. Okay, that is, you can do 3.4 divided by 10. Okay, one turn is 3.4 nanometer divided by, in one turn 10 base pairs are there. So obviously the consequent difference, that is the distance between base pairs, one base pair to another base pair, is going to be only 0.34 nanometer. Okay, between one base pair, let's say adenine and thymine is here, guanine and cytosine here, it's going to be 0.34 nanometer. Okay, now the plane of one base pair stacks over the other in a double helical. This in addition to the hydrogen bond confers the stability. So hydrogen bond is one of the sole reasons for the stability of the DNA. Okay, hydrogen bond is one core reason for the stability of DNA. Okay, stability of DNA. Got it? All of you? Done. Okay, the hydrogen bond is going to lead the stability of the DNA. So here, when you're writing the salient features, okay, salient features of DNA, remember, first salient feature, okay, is going to be that the DNA is a double helical structure. Double helical structure. Okay, double helical structure made up of what? The backbone is made up of sugar and phosphate groups, and the base pairs, nitrogenous base pairs, are projecting inwards. Okay, projecting inwards. Okay, projecting inwards. Okay, next thing you'll remember is about adenine, thymine, that is your purines and pyrimidines. Okay, binding with hydrogen bond, same thing goes, just write the number of hydrogen bonds and then give a mention about the Chargaff rule. Okay, I'm just telling you how to write, okay, so that you know which are the points to write. Chargaff rule. Then you can write about the helix, you can write about the right-handed fashion. Okay, right-handed fashion. You can write about the turns and how many base pairs are present and what is the distance. Distance question might come, students. Distance between the base pairs, so don't forget, make sure that you write, okay, distance between the base pairs. Got it? This is the few salient features you can remember. Then you can also write that hydrogen bond gives the stability for the DNA. Okay, stability for the DNA. Yes, done. Okay, next topic is about the central dogma. So now that you have understood everything about DNA's general structure, be it how they are formed, be it what is there, now you have to study the central dogma. The central dogma means, what is this chapter whole about, right? What is this chapter focusing on, or what is the main goal of this chapter, not just the chapter, in our body also, what is the central dogma. So see, we all have DNA. This DNA can form mRNA. This DNA can form mRNA. This mRNA can form proteins. This mRNA can form proteins. DNA can form its own thing. That is, DNA can form its own structures, and that is, they can replicate themselves. So that will be called as replication. They can form their own replica. Right. Now DNA forms mRNA. That process is called transcription. When mRNA forms protein, that is called translation. That's called translation. So central dogma means where the DNA forms mRNA, mRNA forms protein. Now when DNA forms mRNA, it will be called as transcription. When mRNA forms protein, it is called as translation. Understood? It's called as translation. Now when DNA can form its own replica, it is called as replication. Now this is the central dogma, or I could say this is the ruling factor of your entire molecular biology. The entire chapter revolves on this topic. At the same time, you should understand that the entire life revolves on this topic. Then this protein will be able to, there will be genetic codes, and this protein is going to make the differences or the changes. Okay, so central dogma is a complete ruling factor of this. Now students, have you ever thought, I'll come back to that. Imagine, like, they have introduced central dogma. Now imagine a 2.2 meters of thread. How much? 2.2 meters of thread you have. And I am telling you, you have to place it inside a small nucleus. I'm telling you, you have to place it inside a small nucleus. You'll be like, ma'am, are you crazy? We cannot do that. How can I make a 2.2 meter long thread inside a small nucleus, right? You'll be shocked. So even when the DNA is formed, this question started happening. 2.2 meters of long DNA, a thread-like structure called DNA, has to be fit inside the nucleus. How is this possible? That is when people understood there is something called as DNA packaging. Okay, there is something called as DNA packaging. Students, our body is amazing, and it is crazy that these systems are there. Okay, now packaging means what? See students, all of you have seen how a chain will be formed. Okay, I do not have a chain like that, but if you've all seen those beaded chains, how do you see the chains? There is a bead, then there is a small portion of the chain, again there is a bead, chain, bead, chain, bead, chain. Same way your DNA will be bound. I'll tell you how. This chain goes around the bead. Okay, this chain goes around the bead. Same way this chain goes around the bead. This chain goes around the bead. Like this, it will keep going around the bead. Okay, now this 2.2 meter of DNA will be wound around, or it will go around a bead-like structure or a round circular structure called the proteins. Okay, proteins. So there are some exceptional proteins. These proteins are called histone proteins, which will help in the packaging of DNA. So let's take that these histones are of 4 histones will be there, who's going to come for the packaging. That is H2A, okay, H2A, H2B, H3, H4. Okay, H4. These 4 histones are going to help in the packaging of DNA. Let's first take these histones. Okay, let's just first take these histones. Okay, this is histone number 1. Okay, this is histone number 1. This is histone number 2. This is going to be histone number 4. And this is going to be histone number, this is the histone number 4. Okay, these are the histones. Now let's go back to making it a thin line, and remember each histone will come, 2 people will come. Okay, 2. This is also going to be 2 in number, this is also going to be 2 in number, this is also going to be 2 in number, this is also going to be 2 in number. So 2 people will come. Okay, This one also will have a, I'm trying to draw 3D, okay, possibly it looks like a 3D, okay, and then here also we'll have two people in the back side, okay, two people, and then it'll like form like a cube, okay, it'll form like a cube, two people. So students, now you can see there is four people, right? Four people. This four people, I'm going to call it as nothing but histone octomer. What will I call it? Histone octomer. Octomer means what? 8 structures. Now, how the DNA is going to go around it? That is a question all of you will have. Let's take which color for DNA? Blue color. Okay. DNA. Students, remember that the DNA structure is going to go around like this. Okay. Going to go around like this. Going around like this. Okay. And coming around like this. So the DNA is going to go or wound around this histone. So like this, remember that okay, how this whole structure is going to be like. There's going to be one histone, there's going to be another histone, there's going to be another histone, there's going to be another histone. Okay, so this DNA is going to be wound around these people like this. Okay, wound around these people like this. It will keep winding around like this, and then it will become condensed. Okay, it will become condensed to form chromatin fibers. Like this condensation will happen. Okay, and then this condensation will be followed by the formation of a clean sister chromatids. Okay, so the ones that you study in your prophase, metaphase and all of this, no, that is a combination of many of these histones, students. Okay, many of these histones together. So if I'm drawing here, okay, this is one, this is one, okay, this is one, this is one, this is one, and then they become, you know, condensed, and they will form. Okay, they are going to form condensed structures, and that's how they become this much, you know. So they'll go like this, they'll go like this, they'll go like this, they'll go like this, and it is going to condense around and form like this. Understood? So now this entire thing is called histone octomer. But when the DNA strands, okay, when the, so I'm erasing this, when there is a DNA strand plus the histone octomer, okay, remember, DNA plus histone octomer together is going to form your nucleosome. Is going to together form your nucleosome. So DNA plus the histone octomer is together going to form your nucleosome. Now students, imagine like DNA is put, okay, one pearl is there, thread you have put, but don't you think this thread can come out at any time? So to keep all of this together, okay, to keep all of this together, we have another protein, okay, from the histone itself called as the H1 protein. What is it called? H1 protein. This H1 protein can also be called as the linker protein. Can be also called as the linker protein. Linker protein means it keeps everything in place. Okay, it keeps everything in place. So there are 8 structures which is called as histone octomers, that is H2A, H2B. So all this together will form 8 structures called as histone octomer. Okay, histone octomer. This entire histone octomer will form with the DNA will form the nucleosome. Many nucleosomes like this, many nucleosomes together will form the chromatin. Okay, that chromatin will again condense. Okay, here all condensation is going to happen. Again condensation is going to happen to form chromosomes, and then this chromosomes will enter inside the nucleus that I asked you in the starting. No, they will enter inside the nucleus, and that is how the entire 2.2 meters of DNA is inside the nucleus. Without consuming a lot of space, these people are be able to be inside the nucleus is because of this nucleosome, this histone octomer packaging. Okay, this histone octomer packaging. I hope this is clear to all of you. So let's just see here what they're trying to say. See, taken the distance between the two consecutive base pairs as 0.34 nanometer, that is 0.34 into 10 to the power 9 minus 9 meter. If the length of DNA double helical is typically mammalian cell is calculated simply by multiplying, we are going to get around 2.2 meters. How we are going to calculate? 6.6 into 10 to the power 9 into 0.34 into 10 to the power minus 9. Okay, this and this will get cut, and this if you multiply, you will get somewhere around approximately 2.2 meters. Okay, this 2.2 meters is a length that is greater or far greater than the dimension of a typical nucleus. Typical nucleus approximately is going to be 10 to the power minus 6 meters. Okay, 10 to the power minus 6 meters. So how is such a long polymer packed in a cell? This is the question that arose. Okay, this is the question that arose. So in eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. Okay, histones. A protein acquires charge depending on the abundance of amino acid. So the abundance of amino acid gives them the positive charge. Since DNA is negative, it will be easy for them to wound around the positive charge. Okay, now histones are rich in basic amino acid residues like lysine and arginine. Okay, this you have to remember very importantly, which is the basic amino acid residue that is high in your histones. It is lysine and arginine. Both the amino acid residues carry positive charge in their side chains. Histones are organized to form a unit of 8 molecule called histone octamer. Okay, histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as the nucleosome. A typical nucleosome consists of 200 base pairs of DNA helix. Very, very important. A typical nucleosome, okay? A typical nucleosome contains 200 base pairs, okay, of DNA helix. So nucleosomes constitute the repeating unit of structure in the nucleus, which is called as chromatin. I already showed you, thread-like stained color bodies and seen in the nucleus. The nucleosomes in the chromatin are seen as beads on the string structure. So students, if let's say this is the microscopic, let's say this is the microscopic view. Okay, microscopic view. Then students, microscopic view. So under the microscope, if you see, the beads are there, strings are there. Beads are there, strings are there. Beads are there, strings are there. Now beads, what am I telling? Those are the nucleosomes, right? Those are the nucleosomes. So like this. So this bead on a string structure. Bead on string structure. Okay, bead on the string structure. This bead on the string structure, you can see it under the microscope. Why? Because these are the nucleosomes, these are the DNA strands going around, and then they are rotating. Okay, this is the bead on the string structure, which you can see under the electron microscope. Okay, students, we will resume back. Okay, we'll go for a short lunch break. So resume back in okay, we'll resume back in 15 minutes. So that's a short break. See you all after the break. Okay, see you all after the break. Hello students, let's continue the class. The break is over. I hope all of you had your food as well. Okay. Yes. So we have understood up to the salient features. We have understood how the packaging of the DNA happens. Yes. What happened to this? Okay. Yes, how the packaging of DNA happens. Now we have to learn about some euchromatin and heterochromatin, students. So look here, in the DNA there are going to be regions which have some dark regions, which have some light regions, but other than that there is something called as non-histone chromosomal protein, NHC. That is, when the histone proteins cannot bind anymore, we will require an additional set of proteins. We will require an additional set of proteins that will help us for the binding, especially in your eukaryote. So look here, non-histone chromosomal proteins in a typical nucleus, some region of the chromatins are loosely packed and are referred to as euchromatins. So we'll come to that. Here, look here, this is the definition. The packaging of chromatin at higher levels require additional set of proteins that are collectively called non-histone chromosomal proteins. So other than the proteins, histone proteins, we have to use some additional proteins, okay, when we have to do the binding, that is called as non-histone chromosomal proteins. So in a typical nucleus, now you listen carefully, some regions of the chromatin are loosely packed, okay, stains lightly, and are referred to as euchromatin. The chromatin that is more densely packed will obviously stain darker, that is called as heterochromatin. Euchromatin is transcriptionally active. Heterochromatin is transcriptionally inactive. That means during transcription they are not very active. So here you have to write this main things. Okay, so it is euchromatin. Okay, euchromatin is going to stain lightly. Why is it going to stain lightly, students? Obviously because it is loosely packed, right? Loosely packed, and they are transcriptionally active. Transcriptionally active. Now what about heterochromatin? Heterochromatin is going to be densely packed. Okay, densely packed. They are going to stain dark, because something is tightly packed means if I pour a little stain also, they'll all get very dark because they are all closely packed. But loosely packed is not like that. No, it's just everything is loose. So in a room, let's say 50 people can get in, but only 10 people are there. Now the chairs are all going to be empty, right? So the class also is going to be very, like, no noise will be there. So like that, same way, light, loosely packed, transcription active. This is going to be transcriptionally inactive. So even if you learn one, you can write the other one just the vice versa or the opposite. Okay, inactive. This is inactive, this is active. Done. Now we'll go to one of the experiments that proved that DNA is the genetic material. Okay, DNA is the genetic material. 1 second, students, here. So search for genetic material kept on happening. So always remember that these as a chain, okay, chain of things. Why I'll tell you it's a chain of things is because voice is clear. Even the discovery of nuclein by measure and the proposition of principles of inheritance by Mendel were almost at the same time, but that DNA as a genetic material took a long time to discover. Now how do I tell? See, now I already know it, okay, but how do I tell that DNA is the one who is doing the transformation or who is doing the inheritance from the parent to the offsprings? How do I know it? I know it because it's now already found, but back then they did not know. They thought, okay, maybe it could be protein, maybe it could be RNA, maybe it could be something else that is transferring this information from the parent to the offspring. So they kept on doing one after the other. Experiments came after experiments came, and they were hunting for the answer. Okay, they were hunting for the answer. In Okay, in 1926, the quest to determine the mechanism Okay. In 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, and Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosome located in the nucleus of the most cells. Okay, so this is all you don't have to know, students. First, you have to know Griffith's experiment. So the experiment was very simple. What did he do? He took Streptococcus pneumoniae. Okay. Streptococcus pneumoniae. And the Streptococcus pneumoniae had two strains, S strain and R strain. S strain is also called as the smooth strain. Okay, S strain is also called as the smooth strain, and R strain is called the rough strain. Smooth strain we are calling why? Because they have a covering. Okay, they have a mucopolysaccharide covering which makes them smooth. Why are we calling it rough strain? We are calling it rough strain because of the they don't have a proper surface. Okay, no proper mucopolysaccharide there. Now students, this smooth strain is virulent. Smooth strain is virulent, and this rough strain is non-virulent. Means does not cause disease. Virulent means able to cause disease or will cause disease. Okay, rough strain does not cause disease. Now what did Griffith do? So Griffith's first setup. Okay, first setup was that he took a mice, he injected the S strain. Now what do you expect? Obviously the mice is going to die. The mice died. Why? Because S strain is virulent, it can cause disease. Okay, second setup he did mice plus R strain. He had to check if this is virulent or not. So he did it. The mice live. Okay, then he thought, let me do one thing. Let me do some masala. So he took mice plus he did heat killed S strain. That means he tried to kill the active part of this S strain that was making it virulent. Okay, and he heat killed the S strain and the mice lived. Then he was like, okay, let me do one thing. Let me take both of the positive results. There is this positive result that let the mice live and this positive result that let the mice live. Okay, that is, he took the mice plus he did a very, very important thing. He did heat killed S strain plus R strain. And for a shock, students, what you would have expected, tell me. What you would have expected, you tell me in the comments. Okay, that ma'am, both the results were positive, right? Even in the mice plus R strain was living, mice plus heat killed S strain was okay, like both were alive. But when they mixed these two results, right? When they mixed these two results, they noticed that, or Griffith noticed that the mice died. The mice died. He was shocked. He was shocked because he was expecting that the mice would live because that was the result expected, right? That was the result. But he understood that something from the S strain got transformed. Okay, got transformed to R strain. Something from the S strain got transformed to the R strain that made R strain suddenly virulent. Make sense, no? Yes or no? Now he did not know who transferred, what transferred, so he simply called this transfer as the transforming principle. He called it as the transforming principle. Got it? Yes or no? He called this as the transforming principle, telling that something got transformed from the S strain to the R strain, which I am not sure what it is. So I will just call it as the transforming principle. Then many scientists came after him. Okay, and when did this happen? In 1982 it happened. 1928 it happened. Okay, Frederick Griffith in a series of Streptococcus pneumoniae witnessed a miraculous transformation. Okay, you do not have to go through the same thing what I told. S strain injected into the mice died. R strain injected into the mice lived. Okay, mice lived. Now here, S strain injected heat kill. He noticed that the mice lived. Then he did heat kill S strain and R strain, the mice died. So this was the reason for the transforming principle. That's when they understood that, oh my god, something is there that got transformed and that did not die with any heat. Okay, that got transformed and that did not die with any heat. Okay, so he called it as the transforming principle. He called it as the transforming principle. Understood? He called it as the transforming principle, and that transforming principle, people believe that it is because the smooth strain. Okay, they believe that the smooth strain has a polysaccharide coat. Okay, they believe that the smooth strain has a polysaccharide coat, and that polysaccharide coat is the one that is making smooth strain virulent. Got it? There is something that made the smooth strain virulent. Understood? This was the first experiment. Now students, we'll finish, we'll do one more experiment and then we will stop. Okay, next one, what happened? This, everyone were talking about the transforming principle, no? Everyone were like, oh, this transforming principle, who it could be. Then three scientists came called as Avery, MacLeod and McCarty. Okay, three people came called as Avery, MacLeod and McCarty, came behind the transforming principle. Okay, came behind the transforming principle. Okay, transforming principle, and they were like, we will find the biochemical, we'll find the biochemical nature of this transforming principle. For that they did three setups. Okay, for that they did three setups. They took so students, from the heat killed S strain, that combination, no, the combination is heat killed, okay, plus R strain. They took this combination and they were investigating on three people. Okay, first thing was they took this strain and they added protease to it. So when you add protease, what is going to happen? The protein is going to die, right? The protein is going to degrade. The protein degraded and they noticed that nothing happened to the mice. The mice died. Okay, nothing, no, the mice died. Okay, so I'll just write here this circle. Okay, the circle is this combination. Okay, now next setup they took was, they took the next setup, the combination, plus they took RNAs, and they noticed that is where the RNA would not be there. Okay, RNA got degraded and they noticed that the mice still died. So what are they trying to say? They're trying to say that when we added protein, the protein got degraded. So protein is not the thing that got transformed from the S strain to the R strain. Okay, then they took the combination, they added RNAs which will degrade RNA, and then they noticed that even RNA is not the transforming principle. That means from the S strain to the R strain, the RNA is not the one who is getting transformed. Then their last try was your DNA. Okay, they took the combination plus they added DNAs. Okay, and they noticed that the DNA got degraded. Okay, students, remember ACE. Okay, this protease, RNAs, DNAs are enzymes. Okay, enzymes which will degrade the protein code, RNA as well as the DNA. Here what happened? The mice lived. The mice was alive. This showed that DNA is the transforming principle. So when DNA was not there, the mice lived. That means what? The virulent character of the S strain did not get transformed to the R strain. Both were non-virulent. So DNA is the transforming principle. But the problem is, this experiment had a lot of drawbacks. lot of drawbacks, in the sense people were not ready to accept this experiment. People were like, no, this experiment does not make sense, or there's no clarity of this experiment. It could be anything like a mixture or something. Like, people just kept telling that this is not a great discovery to confirm that DNA is the transforming principle. So the experiment was done by Avery, McLeod and McCarthy in 1933 to 1944. The genetic material was thought to be a protein, so they worked to determine the biochemical nature of transforming principle. They purified biochemicals, that is proteins, DNA and RNA, from the heat kill S strain to see which ones could transform the live to the R strain. Okay, they discovered that DNA alone from the S bacteria caused the R bacteria to become transformed. So they also discovered that the protein digesting enzyme and the RNA digesting enzyme did not affect the transformation. So the transforming substance was not protein or an RNA, but it was a DNA. But the conclusion or the experiment was not a success. It was a fail. And why am I telling it's a fail? Because many people did not believe it, or many people did not follow this or make it as the last experiment to prove that DNA is the transforming principle, because of which another experiment had to come. That was a Hershey and Chase experiment, which I'll be doing next class. So I hope all of you like this video about molecular basis of inheritance. Today we understood the introduction of the chapter, where we understood what is DNA, what is nucleotide, how does it form a chain, and what are the components of the nucleotide. It is your nitrogenous base, pentose sugar, phosphate group. Under nitrogenous base you studied purines and pyrimidids. You studied that under purines you have adenine and guanine. Under pyrimidids you have uracil, cytosine and thymine. Uracil is only present in RNA, not present in DNA, and it is replaced by thymine. Okay, and then we also learned that adenine always binds to thymine to 2 hydrogen bonds, cytosine always binds to guanine with 3 hydrogen bonds, and these hydrogen bonds make sure that the DNA are in a double helical structure because they are projecting inwards, and the backbone of the DNA is sugar and phosphate group. This is what you learned. Then you also learned the salient features of DNA. You understood what is the central logoma, where DNA becomes mRNA is called transcription, mRNA becomes a protein is called translation, and DNA can form its own replica, so it's called replication, or DNA forms its own copies, it's called replication. Then we came across salient features, done. After that, we understood how the 2.2 meters long DNA is packed inside a small nucleus, okay, which is around minus 10 minus 6 meters. So we understood how small the nucleus is and how do we fit this. We did the method of packaging of the DNA with the help of histone proteins, which are positively charged proteins, which are the basic amino acids which are rich as lysine and arginine. Now around this positive protein, the negative DNA is going to bound around and they're going to be fit inside the nucleus. This way what happens, the entire 2.2 meters of DNA will be packed inside the nucleus, and this will form this histone octamer plus DNA is called nucleosome. Many nucleosome together forms a chromatin. Chromatin will again condense to form your chromosomes, and these chromosomes is what you see in the, inside the nucleus, okay, that you see under the microscope during your prophase, metaphase, anaphase and all of that. And then we understood what is non-chromosomal protein, that is extra additional set of chromosome protein that we need for higher packaging. And then we learned about euchromatin, heterochromatin. Euchromatin is loosely packed, lightly stained and is transcriptionally active. Heterochromatin is darkly packed, densely stained, and it is transcriptionally inactive. Then we study, students, what are the experiments that proved that DNA is the genetic material. The first ever miraculous change that happened was when Griffith was doing the experiment with Streptococcus pneumoniae with 2 strains, S strain and R strain. Out of this, S strain was virulent, R strain is non-virulent, and when he was doing it on the mice, he noticed that there was something that got transformed from the heat kill S strain to the R strain, which he called it as the transforming principle. Then Avery, McLeod and McCarthy checked the biochemical nature of this transforming principle, where they took protease, RNAs and DNA and they degraded it, and they noticed that DNA is the genetic material, because in protease and in RNAs they did not see any kind of transformation. The mice still ended up dying because DNA is still present. Okay, DNA is still present. And then in DNAs, they noticed that the mice lived, that proving that DNA is the transforming principle. So I did a 3 minutes recap of the entire topic that we did today. I hope you really enjoyed this video. Thank you so much for joining. See you all in next class.